Okay, this is the Professor, CBMD PhD, and today's Saturday, October 7th, 2023. It is 2:32 and 20 seconds, AM Eastern Standard Time, and today's research, Glioblastoma, Therapies. So let's hop to it here. Advertise the help, tailored glioblastoma therapies. Date October 4th, 2023. Source, Mass Debra, Center for Molecular Medicine. As scientists have created a new zebrafish xenograft platform to screen for treatments for an aggressive brain tumor called glioblastoma, according to a new study. These patients are really in need of new therapies. It is very important to identify the patients who do respond to a specific treatment and the ones who do not. The scientific investigators and microbiologists have created a screening platform that could be refined to find targets for drugs against glioblastoma. It can also be used to check if a particular patient will respond to a therapy. And to understand how macrophages can interact with glioblastoma cells of different patients, the researchers created zebrafish avatars. The research laboratory works extensively with zebrafish. The scientific researchers investigated glioblastoma stem cells from 7 patients collected by scientists at the laboratory, which is establishing a living tissue bank of glioblastoma samples. The microbiologists injected them into zebrafish embryos, creating xenograft models, an avatar for each specific patient. When they live imaged the embryos, it appeared that the glioblastoma cells had adapted well to their new environment. They saw the zebrafish's immune system sending macrophages as part of an immune response to control the tumor, but as is typical in glioblastoma, the macrophages were suppressed. The tumors have several mechanisms to reprogram the macrophages so they help them grow. They wanted to learn how to revert the macrophages to a tumor-attacking state, and a clue surfaced when they noticed that the tumor of one patient did not suppress the normal macrophage response. Upon closer investigation of the medical details, they discovered that this patient was what they call a long-term survivor. It is a term used for glioblastoma patients with a survival of more than 5 years, which is exceptionally rare in this brain cancer. Their curiosity about the patient became the driving force behind the project when they cultured the tumor cells and macrophages together and did single-cell RNA sequencing. They learned that one gene, LGA-LS1, was downregulated in the tumor of the long-term survivor compared to others. Earlier studies have also shown that silencing of LGA-LS1 and glioblastoma cells can result in longer survival. The scientific investigators confirmed the results by knocking out the gene in another patient sample and observed in the zebrafish models that the tumor became less invasive. As this platform could be used to identify promising targets other than LGA-LS1 for this treatment of glioblastoma, and with this refinement, zebrafish avatars could be used to identify which treatments will work. Researchers could investigate whether the tumor cells from particular patients grafted in the zebrafish respond when treated with various drugs to find the ones that lead to tumor regression. The scientific investigators, the microbiologists say, armed with this information, they can inform oncologists and help them to make more supported treatment decisions for the patient. And this is the Professor of Microbiology, Bioinformatics, Pathophysiology, and Leader of Research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.